thank you, Khaled, for the invitation, and thank you for changing the timing of my talk so I can present earlier as I'm leaving today to Egypt. Okay, I'll talk louder a little bit. So I gave this presentation at KSCC 2 months ago, and my boss came to me and said, I congratulate you for standing in front of everyone and talking about something you know nothing about. So I'm going to repeat it today. Somehow Khaled heard about it and asked me to do the same thing here, so I'm just doing it again. If I show you this sequence of bases from DNA and ask anybody here to know where this is coming from, I think you will immediately realize that no human brain can really analyze this or know what this is all about. And then I'll ask, what branch of science generated the highest number of computer algorithms in history? Is it space navigation or transportation industry or stock markets, or is it bioinformatics? And obviously it is bioinformatics. So this is a field of science where computers play a major role. But it is still a branch of biology, which is concerned with the acquisition, storage, and analysis of sequence data from DNA and proteins. So the most important word here is biology. Because as physicians, as oncologists, we are biologists by virtue of our work. This field is becoming very important for us. You don't need to know much about biology, but the central dogma of molecular biology, which tells us how DNA replicates and then transcribed to form RNA, and then this is translated to proteins. Really, and this is the simplest form of the central dogma, but really this is what this story is all about. Interestingly, in eukaryotic cells and in human genome specific, the process of splicing, taking out intervening sequences from the DNA and building mature messenger RNA is very interesting, especially that you may have different combination of exons to form different transcripts. So every gene has 9 possible, in average, 9 possible transcripts. So that gives diversity to the genes that we already have. And then you're all aware of the changes that may take place in the genome, insertions, deletions, and substitutions. And because substitutions are very common, and we are in a way different from one another, there's something called Snips, which are single nucleotide polymorphism. And this is or now called also SNV, or variation. So now when you have a substitution of one of the bases, this SNP may take place within the coding region of the gene and may be silent, or coding SNP, which may change the structure of the protein. It may affect the promoter of the gene and thus affect the quantity of the gene, and it may occur outside the promoter and still can regulate the quantity or function of the gene expression. So how do we sequence DNA? Because this is really what we need to do. Here I introduced Dr. Frederick Sanger, who got his first Nobel Prize for sequencing bovine insulin, which was available commercially at the time. And then he went on and discovered the Sanger's methodology of sequencing. And in summary, you have fluorescent nucleotides which have terminators. So a PCR reaction will copy a sequence of DNA and produce random length fragments of DNA. And when one of these bases is stuck here, the reaction will stop. The length of the fragment will tell us the location of the base, and the color of the fluorescence will tell us the actual sequence. So this is the first generation genome sequencing, and he got another Nobel Prize for this, and still considered the gold standard for sequencing. But things moved with the next generation sequencing when we were able to do faster sequencing, and the whole human genome sequencing now takes on a machine maybe 1 or 2 days. And the principle here is that we prepare libraries of what is called cDNA, and then a PCR reaction takes place, and then the nucleotides, which has also terminators, are added, and then the reaction stops. A camera will detect the color, and the sequence will be known, and then the terminator will be removed, and the reaction will continue on and on and on and on until we have a read. And each read of these is 150 to 300 bases. So we are not sequencing the whole genome, we are building reads. So this concept is very important to understand the next generation sequencing. This is the machine, and it's a camera. Really what it is, it's a very, very high resolution camera, and it builds lengths of reads, as I told you, in what is called a FASTQ format, which is the raw data of the next generation sequencing. Now, what we did exactly is that we shredded the DNA. We converted the whole DNA to a very large number of reads. And to build the DNA again, we need to put these reads again. Imagine if you have this huge puzzle, but you don't have the picture, and you need to put it together to build the picture. It's very, very difficult. If you have the picture that you are trying to build the puzzle for, things become very, very easy. So there are two ways of building genome. First time we build the genome is called assembly. So we have to put the puzzle pieces together without knowing how this fits. And then there's something called alignment, when you know exactly how the genome, and what you need only to do is to get these reads and fit them where it is, where they fit in the genome. So this is much easier, doable, and this is how things work nowadays. It's something like this. You are trying to put the genome together, the reads together, building something that makes sense out of these, trying to fit these reads in a way that makes a sequence, like this. But in low complexity regions of the DNA, things are not that easy because it might be difficult to build the genome. And for that reason, we still have in the human genome areas that are not really well understood, and we still don't know the whole sequence of some difficult low complexity regions of the human genome. Two terms are important, are the coverage, which is the percentage of the genome that is covered by the reads, and the depth, which is the number of reads that cover each region, which will give more and more accuracy to this sequencing process. This project, the Human Genome Project, started in 1990, and it was planned to go for 15 years. Dr. Francis Collins was the NIH representative of the Human Genome Project, and for 10 years, the project did not really move. Maybe 10% of the work was done. And then the private sector joined, and there was some competition between the NIH and a private company to sequence the DNA. And in 18 months, almost 90% of the work was done. And then we know these facts about the human genome. The project was announced completed in 2002, but really the project will never finish. I think we will never have a full sequence of the human genome, as I said, because of the low complexity regions. So we have 3 billion bases on the human genome. There's 99.9% similarity between different individuals. 2% of the genome is coding and represents genes, and more than 98% is dark matter or non-coding genome. There's a little bit over 20,000 coding genes and 22,000 non-coding genes. The transcript number is around 200,000, which is 9 transcripts per gene. The number of proteins are 30,000, and there are 2,500 mRNA or microRNA, which also are very important biologically. The average human genome is around 1,300 bases and code for 450 amino acids, and has an average for exons. And because we are 99.9% similar, that means in the 3 billion bases that we have, we might be different. Each one of us might be different from the group in 3 million Snips. And these data were published in Nature and Science on the 15th and 16th of February 2002. Since that time, we started a new era in science. And as the NGS became commercially available around 2006 and 2007, the price of doing a whole genome sequencing dropped down, and now maybe you saw this figure, before, but it should be around $1,000 to sequence a whole human genome, which is very, very cheap. But this created a whole lot of problems really for us as clinicians who have to deal with the consequences of this high throughput sequencing data. First, we need some validation, because now we have these many SNPs in each one of us, and then we have the phenotypes that are available. So how can we get the phenotypes linked to genotypes or to these SNPs or mutations or alterations? This is very challenging. The cost of doing these tests maybe is dropping, but the cost of analysis is still high. The complexity of analysis and the need for software is really a challenge. There are many ethical problems. Privacy is a big deal. Who wants his whole genome with all the secrets of your future diseases and your problems that you or your siblings or might have? This is a big problem. And then how can we deal as clinicians with unanticipated findings? If we know that a person has a mutation in a gene that may predispose him or her to a certain type of cancer, for example, how can we deliver this information? Should we deliver this information, and how we handle it? And then there are parts of there are some changes in the genome that are not fully covered by NGS, like deletions and duplication. I'll talk briefly about that, but NGS is not the ultimate answer for everything. We may still miss some changes. And then the number of genetic counselors and geneticists did not increase really with the data that we have. What we have today is beyond imagination in a way, but still the number of people who can deliver this information to patients is limited, and that's really a big challenge. I'll talk briefly about the applications of NGS that are pertinent to oncology. First, we do something called variant calling, which is identifying the changes that take place at a certain loci. And from that, as I said, we may want to link these changes to phenotype, and this is very challenging without the collaboration of many institutions and open repositories, and even patients in platforms like MyGene2, who may enter their phenotypic data and help reaching out to families and physicians who might be interested in very rare diseases like the ones we just heard about. This platform and collaboration help tremendously in understanding and diagnosing more and more genetic disorders. We still have 1/3 of Mendelian inherited disorders with no link to specific genes, but the number of diseases that are known or maybe have some what's called phenotypic expansion, which is understand more and more about the different phenotypes of the same disease by linking different SNPs that might be away from the certain gene to the disease. Skip this, and then the Genome-Wide associated study. This is very important, especially in cancer. As I said, the changes may not really take place within the gene. We may have changes outside the gene, and for the common diseases like hypertension or diabetes or cancer, most of the changes really that may predispose individuals to cancer are not within coding genes, but are rather within non-coding areas of the genome. And if we look at certain SNPs at a certain location, compare tens of thousands of controls to cases, and then do statistical analysis, we get something which is called Manhattan plot, and these spots represent very low p-values, and thus then we can understand on the genome where this is located and know how to link a certain SNP to a disease like cancer or diabetes, as I said. And then in cancer, we are very interested in mutational burden, because that tells us if the disease is susceptible to immunotherapy, which is becoming more and more important. In pediatric cancers in general have low mutational burden in comparison to adult cancer, with some exceptions. Then we have copy number variations, and that's also very important for us. We may have duplications or deletion of a certain block of genome. And this can be analyzed by NGS as well, because the NGS does not only give us a qualitative description of the sequence, but also it quantifies the number of the reads. So in this example, if we have high number of reads in a certain area, we know that this represents amplification or gain of a gene. And this can be done for a high number of patients and studied, like this study which looked at leukemia, at copy number variations in patients with leukemia before and after relapse. And you can tell that there's some correlation in the copy number amplification in red or deletion in blue between the samples on the left that are the original samples and the relapse samples. So this kind of research is really can give us amazing results. And this really what got me interested in this field, because this is how we do cutting-edge research using low budget methodologies. Structural changes like inversion and translocation are very important, especially for pediatric cancers, namely pediatric leukemias. And these are really important, but I have to clarify that the NGS methodologies are not robust for many of these. So till today, we depend on cytogenetics to diagnose these alterations. Epigenetic changes are important, and I cannot really emphasize that enough. As you know, we have one genome. Each one of us have one genome, but we have millions or billions of epigenomes, changes that take place over the genome, namely methylation or histone modifications like acetylation and methylation. So these changes may change the function of the genes by making certain areas of the chromatin more open for transcription or more close and wrapped around histones or nucleosomes. A fascinating platform was developed by DKFZ in Germany, and what they did using artificial intelligence is to take those methylation studies from thousands of patients and study the changes that take place in different brain tumors, pediatric brain tumors namely. And doing this methodology, they were able to develop an algorithm that will diagnose patients without histology. So now you submit the methylation study to this website, and they will do for free analysis and give you the diagnosis without the help of a pathologist. And if any one of you is involved in this, you will understand how difficult this is. You know, neuropathology, especially pediatrics, is extremely challenging. And if we can do this just by doing a methylation study, this is a new field of medicine, and maybe pathologists will be the first, the first career that will disappear in the new era of artificial intelligence and high throughput sequencing. And this is how it works. It separates patients into groups based on methylation, as I said. RNA-seq is extremely interesting, and especially for cancer. What it is, imagine if we have patients with certain diseases, or maybe before and after treatment, and we want to measure the protein alterations that take place before and after we give a patient a treatment. If we want to measure protein levels in patients before and after, that would be very expensive because we need to do tens of thousands of protein assays. But if we measure the RNA changes that take place before and after maybe treatment, or we compare patients with non-metastatic disease with metastatic disease, we know which RNAs are involved in the process, and this can help in understanding the pathways that are involved in this. The process is computationally very challenging because we need to reconstruct RNA, which is not easy. As I said, there are different ways that exons attach to each other to make transcripts, but the end result is amazing. You get something called expression table with levels of each RNA in each individual, and then you can compare for each gene the levels in different setups. And because we're doing this for 20,000 genes, it's hard to visualize this or understand it without a volcano plot. And here, the far away the gene is from the midline, the more fold change is taking place, and the higher the level is, the lower the p-value, which means the more significant change. And then we can look really at these genes here for patients with condition like metastatic disease or these for patients with no metastatic disease. I think my time is up, so I'm going to skip some of these slides and go to the last slide just to do some advertisement for our cancer care informatics program that was established at KACC. And in last, I know, this is just a commercial, so it doesn't count. So in last November, we had our first cancer care informatics conference in collaboration with the University of West England, Bristol, and it was really a great success. We had 6 international speakers coming to the conference. We plan to have the conference next November, inshallah. Also, we are starting at KACC Masters in Cancer Care Informatics. We already have applicants, and hopefully we'll start in 2 months this very interesting program. And I hope also we'll have a diploma in bioinformatics that may really help young investigators. I'm sure some of them are here in the audience who might be interested in this work, which as I said, may lead to cutting-edge research really with very limited resources, especially that many of these data is already available online for free to be downloaded and analyzed, and we can learn a lot still about cancer from these data. These are some resources that might be useful for this field, and I stop here. Thank you.